Hello everyone, welcome to Nutrimentor. In the previous session, we covered fats and oil, and in this session, we will focus on important questions from enzymes. Let's begin with very fundamental question. Enzymes are mainly classified as, and the options are carbohydrate, proteins, lipids, and minerals. So we use enzymes every day in metabolism, but what are they actually made of? Are they carbohydrate, lipids, minerals, or proteins? The right answer is proteins. Most enzymes are proteins that function as biological catalyst. So the question is the substance on which an enzyme act is. Options are product, substrate, inhibitor, coenzyme. As you all know, every enzyme needs something to act upon. What do you call that molecule? This term appears repeatedly in biochemistry questions, so make sure you remember it. The right answer is substrate. A substrate is the specific molecule that binds to an enzyme and undergoes a reaction. So let's take another question. Enzyme increase the rate of reaction by. Enzymes make reaction happen faster, but how exactly do they do it? Do they add energy, remove energy, or change the reaction pathway? Think conceptually. So the options are increasing activation energy, lowering activation energy, changing the product, or stopping the reaction. What could be the right answer? The right answer is lowering the activation energy. The enzyme present in saliva. The options are pepsin, amylase, lipase, and trypsin. Digestion actually begins before food reaches the stomach. Which enzyme in saliva start breaking down carbohydrate right inside the mouth? And the right answer is amylase. Salivary amylase begins the digestion of starch in the mouth. Pepsin is active mainly in and the options are mouth, stomach, small intestine, liver. So different enzyme works best in different organs. Pepsin is famous digestive enzyme, but where exactly does it do its job? The right answer is stomach. Let us take next question. Lipase is responsible for digestion of proteins, fats, minerals, and vitamins. A quick digestion question it is. If fat needs to be broken down into smaller molecules for absorption, which enzyme is responsible? Recollect your biochemistry. Yes, the right answer is fat. Lipase breakdowns dietary fat into fatty acids and glycerol. A coenzyme is generally derived from vitamins, proteins, carbohydrates, and water. Enzymes often need helpers to function efficiently. These helpers are called coenzymes. But where do most coenzymes come from? Yes, you guessed it right. The right answer is vitamins. Many coenzymes are derived from B complex vitamins. The enzyme that digests starch in small intestine. Options are pepsin, pancreatic amylase, maltase, renin. Starch digestion doesn't stop in mouth. Which enzyme takes over in small intestine and continues carbohydrate breakdown? Give it a thought. Yes, the right answer is pancreatic amylase. Pancreatic amylase continues starch digestion in small intestine. Enzyme specificity refers to acting on all substrate, acting on a specific substrate, acting only in stomach, acting without substrate. So one enzyme cannot work on everything. You all know that. Why is that? This is important property explains why enzymes are highly selective. So the right answer is acting on a specific substrate. Enzymes usually act on one substrate or a small group of related substrate. Of inhibition, the effect of enzyme kinetics is Km increases, Vmax unchanged. Km unchanged, Vmax decreases. Both Km and Vmax decrease. Both Km and Vmax increase. This is one of the most important conceptual questions in enzyme kinetics. When a competitive inhibitor is present, what happens to Km and Vmax? Think carefully. The right answer is Km increases, Vmax unchanged. Competitive inhibitors compete with the substrate for the active site. The protein part of an enzyme without its cofactor is called holoenzyme, apoenzyme, coenzyme, and zymogen. So, an enzyme isn't always complete on its own. If the protein portion is present, but the cofactor is missing, what do we call it? The right answer is apoenzyme. Apoenzyme is the inactive protein component of an enzyme. So, an inactive precursor of an enzyme that requires activation is called apoenzyme, isoenzyme, zymogen, and holoenzyme. Some enzymes are produced in an inactive form to prevent damage to the body tissues. What is the scientific term for such an inactive precursor? So the right answer is zymogen. Zymogen are inactive enzyme precursors activated when needed. Enzymes that catalyze cleavage of bonds without hydrolysis or oxidation belongs to hydrolases, lyases, transferases, ligases. Let's test enzyme classification. Which class removes groups and often creates double bonds without using water or oxidation? The right answer is lyases. Lyases removes groups from substrates without hydrolysis or oxidation. Which of the following is coenzyme from vitamin B2? Is it NADP, FAD, TPP, coenzyme A? Vitamin-derived coenzymes are favorite exam topics. Which B2 gives rise to which important coenzyme involved in oxidation reduction reaction? And the right answer is FAD. FAD is derived from riboflavin B2 and participates in redox reaction. The temperature at which enzyme activity is maximum is called. Options are critical temperature, optimum temperature, boiling point, freezing point. So every enzyme has a temperature range where it performs best. What do we call the temperature at which enzyme activity reaches its peak? And the right answer is optimum temperature. It is the temperature at which enzyme activity is found to be highest. Beyond optimum temperature, enzyme activity decreases due to, and the options are increased substrate, reversible inhibition, denaturation of enzyme, change in pH. If the heat generally speeds up reaction, then why does enzyme activity suddenly fall after a certain temperature? What's happening to the enzyme molecule there? And the right answer is denaturation of enzyme. High temperatures usually disrupts enzyme structure and cause denaturation. A complete active enzyme consisting of apoenzyme and cofactor is called apoenzyme, holoenzyme, coenzyme, isoenzyme. When the protein part and its helper molecule come together to form a fully functional enzyme, What is that complete structure called? Give it a thought. Yes, you are guessing it right. The answer is holoenzyme. Holoenzyme is nothing but a combination of apoenzyme and cofactor. Region of an enzyme with substrate bind is called active site, binding site, reaction site, or catalytic zone. Imagine an enzyme as a lock and a substrate as a key. Which part of enzyme actually receives and binds the substrate? Yes, the right answer is active site. It is a specific region where substrate binding and catalysis occur. The lock and key model explains, and the options are enzyme inhibition, enzyme specificity, enzyme synthesis, or enzyme degradation. You've probably heard the phrase lock and key in biochemistry. Which fundamental properties of enzyme does this model help explain? And the right answer is enzyme specificity. The lock and key model illustrates how enzyme recognize specific substrates. The activity of enzymes is highly affected by, and the options are light only, temperature and pH, pressure only, and color. Final question. Two environmental factors can dramatically change or increase or decrease enzyme activity. Can you identify them? The right answer is temperature and pH. They influence enzyme structure and activity. Let's quickly revise the entire enzyme chapter in less than a minute. First, remember that enzymes are biological catalysts made mostly of protein. They speed up reaction but are not used up during the process. How they speed up reaction? By lowering the activation energy, the shortcut that helps reaction happen faster. Salivary amylase starts carbohydrate digestion right in the mouth. Once food reaches the acidic stomach, its action stops. Pepsin is a major gastric enzyme that begins protein digestion and works best in the highly acidic environment. Pancreatic lipase helps digest fat in small intestine by breaking triglycerides into fatty acids and glycerol. Trypsin and chymotrypsin continue protein digestion in the intestine and function best in an alkaline medium. Many enzyme needs helper called coenzymes. Most of these are derived from B complex vitamins. For example, FAD comes from vitamin B2 and NAD comes from vitamin B3. So don't confuse Apoenzyme and holoenzyme. Apoenzyme is the inactive protein part alone, while holoenzyme is a complete active enzyme formed after combining with its cofactor. Digestive enzymes are often stored as inactive forms called zymogens. Examples include pepsinogens and trypsinogen. In competitive inhibition, an inhibitor occupies the active sites and competes with the substrate. The effect can be reduced by increasing substrate concentration. Finally, enzymes activity depends greatly on temperature and pH. Extreme condition can denature the enzyme and permanently reduce its activity. That's your complete enzyme revision. If you can recall these points confidently, you have covered most of the important enzyme concepts for CPGET. Thank you for watching and stay connected with Nutrimentor.